Hey guys, so today we're going to be looking at mitosis and how that relates to cancer. So in order to do that, we're going to work with this online simulation here. That's going to be a microscope looking at some virtual slides that will represent what some normal and cancerous tissues will look like. So as you guys may have already heard, there are a couple different stages of mitosis. We have interphase, prophase, metaphase, anaphase, and telophase. So let's just do a quick review before we get started. So interphase is basically the stage where the cell isn't thinking about reproducing at all. It's just living its best life. It's growing, it's replicating its DNA. It's just doing whatever it needs to do to survive. So in interphase, there's really no reproduction going on. Next, we have prophase. So this is where the cell begins replication. So the nucleus and the nuclear membrane begin to break down and disappear. So you'll be able to see some of the chromosomes. And then the spindles of the centrioles are going to start coming out. So you can see like these yellow lines here. Yep. So the spindle fibers are starting to grow and start moving on either side. And the chromosomes here, we have two blue ones, two red ones. So each of the same color is going to be called a sister chromatid. So moving on to the next stage is metaphase. So metaphase is when the sister chromatids, each of the colors, line up in the middle of the cell. And the centromeres, where the spindles are here, so centromeres right here and right here, is going to come out attached to those chromatids before we go on to the next stage here, which is anaphase. So here, the centromeres divide, and the sister chromatids are quite literally pulled apart. As we can see here, originally we had that X, and now we can see that they're like pulled apart into like these two V's. So from there, these chromatids begin to get pulled away from one another to each side of the cell, and each chromatid then becomes its own separate chromosome again. And then finally, telophase is the final stage of mitosis. So the centrioles and the spindles begin to disappear, and the centrosomes stretch out and become harder to see. And then that nucleus and their membrane starts forming again. And then the two sides are going to start pinching off and creating two whole new cells. And then once that pinch off is completed, then we have completed our cellular replication here. So what we're going to be doing, now that we know what each of the phases are, we're going to be looking at some different slides here. So here we have slides for lungs, stomachs, and ovary, and each one of those body parts has either a normal section or a cancerous section. So we're going to go through each of these and fill out our table in your worksheets here. So we're going to start off with a normal lung here. So under our microscope slide here, this is what we're going to be seeing, and we see a bunch of different cells here. So what we're going to do in these boxes is going to drag and drop the names of the phases that the cell is in. So here we can see there's no replication going on. He's just chilling. So we're going to take our interphase here and we're going to drag it down into our box, and then we'll go on to the next one. This one is also interphase. Since there's no replication, we can't see the chromosomes. There's a clear nucleus. Same here and here. But as we can see the chromosomes here. We can see some replication happening. So what we're going to do is go through these phases and find the one that most resembles it. So here we can see that the nuclear envelope is starting to break down, and we don't quite have the centrioles or the cell splitting off quite yet. So this is going to be prophase. So just going to double check here, and it looks like we got it all right. So now in your table, you're going to see normal lung, and then you're going to see interphase, prophase, metaphase, anaphase, and telophase. So I'll pause here for a second and give you some time to jot down, count how many cells are in each phase, and just jot those down into your table. So in the normal lung row, write down how many cells you count in here that are in interphase, how many are in prophase, metaphase, anaphase, and telophase. So pause the video if you need time to count, and I will see you guys when you're done with that. All right, so right now you should have basically all these cells are in interphase except for our one prophase cell. So again, this was just our normal lung, and now we're going to switch over to our cancerous lung here. So now we're just going to go through the same process here. So let's start right here. We can see again, this cell is an interphase. There's no replication happening, no mitosis. There you can't really see any of the chromosomes, and you can see a clear nucleus. And we say two other cells looking the same in interphase. And now we have some different ones right here. So here we can see that the cell is starting to split off. We can see those sister chromatids being pulled apart. So it's not metaphase, they're still together. And then here in anaphase, we can see that split here. And then here we can see that we see the cell pinching off more. The chromatids are still being pulled to each side of the cell. So this is going to be telophase. Let's check our work. Perfect. So now just do the same thing in your table. Count how many cells are in interphase, how many cells are in prophase, metaphase, anaphase, and telophase in that row that says cancerous lung. Again, I'll give you guys a second to do this. Feel free to pause the video if needed. All right, welcome back. So we just worked with both of our normal and normal and cancerous lung slides. So now let's switch over to some stomach slides here. So again, we're just going to do the same process over again. I'm just going to start with interphase. So we got here, here, here. And then we have metaphase. We know this is metaphase right down here because we still see those X chromatids lined up in the middle, and they haven't been pulled apart yet. And then this next one, we see how the cell is kind of splitting, kind of like that figure 8 shape. We're going to pull it, and then that means that is telophase. And again, here is our results. So again, count how many cells are in each stage of mitosis. And I'll give you a second, pause the video to get that written down. All right, so now we're going to switch to our cancerous stomach. So our cancer stomach, this one looks a little more complicated. So starting off here, it doesn't look like any of our cells that we have to label are in interphase. We can all see the chromosomes in all of them and how they're all starting to line up to some degree. So starting off, we'll start at the very bottom. We have prophase. This is prophase because we see visible chromosomes, but they're not lining up or being split apart yet. Next, do we have any metaphase? We do. We have it right here because we can still see our chromatids are in those X's right in the middle. And then up here, I missed it. We have another prophase. Next, we have anaphase right here. We can see how we kind of have one whole cell and the chromatids are being pulled apart. And then we have finally our telophase. All right, so again, fill out your table. I'll give you a sec and I'll see you in a second. All right, and next we're on to our last type of tissue, ovaries. So this is gonna be our normal ovaries. So I'm just gonna really quick, just rapid fire through this. So anaphase, we got metaphase lined up in the middle there, but not splitting. Clear nucleus without being able to see the chromosomes. And then anaphase, you start seeing some split. All right, perfect. So fill out your table now. And then finally, we're going to go to our cancerous ovary here. And then I'll just blow through this again. Metaphase, telophase, and I'm assuming it's talking about that one there. So we are going to do interphase. And now you guys can fill out your chart completely here. So then once your chart is completely filled out, you guys can go and fill out the rest of the worksheets, questions 1 through 3. So based on your data and observations, what are the difference between the different normal cells and the cancer cells? So make some observations there. And then calculate out the meiotic index. It tells you how to do that right on the worksheet. And then answer question number 3. So if you have any questions, please ask your teacher. They'll be happy to help. And I will see you guys later.